All right. Hello, everyone. Thanks for joining us today for the AIS webinar series. We're excited to have you all here. I'm Megan Weber. I'm an extension educator with the Aquatic Invasive Species Detectors program at the University of Minnesota. And today we're joined with Dr. Amy Kinsley, here to talk about chytrid fungus in amphibians. And it's a fun topic to have for us because I think this marks our first ever amphibian-based topic in the series, so it'll be fun to learn about something new. I have a couple of housekeeping items for us all before we get started. I'm just going to turn it over to Amy. So the webinar is getting started now at 1. We have an hour set aside for this. About half of that will be presentation time, and then we have about half of that set aside for Q&A. We do manage our Q&A through a tool called Slido. You'll be able to access that with either the website showing on the screen now, and then the event code is Amphibians. Otherwise, once it gets to Q&A, I'll also put a QR code on the screen that you can scan with a device to access it. We do have other opportunities if that tool doesn't work for you, or if you'd rather not use it, that's okay. We'll be able to get your questions one way or another. We are also recording today, so if you have to leave early or hear something you want to come back and visit or see something you want to share with other folks, there will be a recording available. We post them up onto our YouTube channel. Just know it takes a little bit of time to get them transcribed before they go live there, but you will get an email notification once that recording is live. Everyone will be muted throughout the event, but if you have questions, again, just use that Q&A feature, or if you have problems during the webinar and need some tech help or something like that, feel free to use that chat function. And my colleague Michelle, who is sitting behind that AIS Detectors program name, will be happy to help you out throughout. That is all I have for the housekeeping items. So I'm going to turn the screen over to Amy to start hearing more about chytrid fungus.
Great. Thank you, Megan, and thank you all for joining today. I'm going to go ahead and get my screen sharing. So today I'm going to talk about a type of invasive species that are so small that we can't see them without a microscope. And so it's what I like to call invisible invaders. So like invasive species that we're familiar with, these organisms can spread through ecosystems long before we have any idea that they're present. And the difference is that these invaders that I'm focusing on today, which are chytrid fungi, are a group of fungal pathogens that infect amphibian hosts, which means that detecting them requires that we find infected or potentially infected animals and then use specialized diagnostic tests to identify if the pathogen is present or not. And so today I'm going to walk you through what we know about these pathogens, what the risk might look like for Minnesota, and how surveillance networks, including those that are student-driven, can help us detect and respond to these threats. But before I jump in the presentation, I want to acknowledge the team that's behind this work. This work is highly collaborative. I work with Jennifer Lamb at St. Cloud State University. She and I lead the project with our colleagues at U.S. Geological Survey, which includes Daniel Greer and Megan Winsler. And then we have a number of fantastic students and trainees who've been involved in the fieldwork and in data analysis. And that includes Galia Motabernia, Emily Banks, Momo Zambalen, and Carrie Beers. And we've also collaborated with the Minnesota DNR, who bring in their expertise in amphibian conservation and knowledge about the presence of amphibians across the state. So to understand why we care so much about these fungal pathogens, we need to zoom way out. And we know that life on Earth is incredibly diverse and that this diversity is what allows our ecosystems to function, thrive, and remain resilient. But we also know that biodiversity is declining worldwide and that many studies have consistently shown that species are disappearing faster than at any time we've seen in recent history. And there's been multiple factors that have been identified that are driving these losses, which include habitat destruction, climate change, pollution, invasive species, which we're all aware of and interested in here, and emerging infectious diseases. And these drivers don't act in isolation. They interact and they amplify with each other. And through, we know that invasive species and emerging diseases are separated a lot in studies, and at times also in their governance and control and regulation. But pathogens that cause emerging diseases are also considered invasive species, especially when they cause significant harm when they're introduced into new ecosystems. And so, although declines that we've seen are multifactorial, one of the most devastating drivers of declines in amphibian populations have been caused by the chytrid fungus Batrachochytrium dendrobatitis, which is a mouthful, so we just call it BD. It has led to large global impacts, and there are estimates that 40% of amphibian species that are currently on Earth are threatened for extinction. They're at risk of going extinct. So our global analysis suggests that BD has contributed to the decline of at least 501 amphibian species, which is quite a bit. And if you look at the figure here, we can see that these declines have occurred across multiple continents. And those that are most severely impacted are in Central and South America. But BD is not the only chytrid pathogen of concern. More recently, a closely related species, Batrachochytrium salamandrivorans, which we call BSAL, has been identified in fire salamanders. That has led to the collapse of an entire population in salamanders in the Netherlands and has led to mass mortality events throughout Europe. And so our current evidence suggests that this pathogen likely has spread through global amphibian pet trade, and that there are some species who can carry the fungus without showing any symptoms at all. And this raises a huge concern for us in North America. We have one of the highest diversities of salamanders in the world. And luckily, so far, current surveillance efforts have shown that BSAL has not been detected in wild populations in North America, but that the risk of introduction remains. So here I'm showing the life cycle of BD to highlight how the chytrid fungi infect hosts and how they spread once they're introduced into a habitat. The fungi produces microscopic swimming spores that are called zoospores, and they move through the water. The spores then attach to the amphibian skin and encyst, and then they begin growing on that outer layer of skin. Within a few days, the fungus then produces new spores, which are released back into the environment and infect additional hosts. So because amphibians rely on their skin for respiration and for electrolyte balance, really heavy infections can disrupt these processes and can lead to mortality. And so the life cycle is very similar for BSAL, which tends to cause more destructive skin lesions. And BSAL prefers salamanders. So of the amphibian groups, it has a predisposition for salamander species. BSAL skin lesions can look like the ones that I'm showing here. They often show up as ulcers or areas of skin damage, but interestingly, these symptoms aren't diagnostic. Skin lesions can occur for a variety of other reasons. And to add more complications to this, infected animals don't always show these skin lesions. So some animals might appear healthy and they're also still carrying the pathogen and potentially spreading to other animals or leading to heavier amounts of zoospores in the environment. And so this is why visual observation alone isn't sufficient. We have to confirm infection with diagnostic testing, which we do with skin swabs and PCR. Once amphibian populations in the wild become infected with chytrid fungi, large-scale treatment is really challenging and has to date not really been effective. Because of that, prevention and early detection are critical. So what I'm showing here are what researchers in Australia have been exploring. They've done some creative work with mitigation strategies that they are calling frog saunas or frog spas, which are these small shelters that allow the frogs to go in and then warm themselves. And the higher temperature can limit the growth of the fungus and allows the animals to then recover. And what's interesting that they've observed in their studies is that sometimes animals who have gone through this infection and survived through these frog spas or frog saunas have been resistant to reinfection, indicating that maybe there's some immunity that exists if the infection does not kill them, and they're allowed to thermoregulate in a way that inhibits the pathogen's growth. So essentially, it's like a way for the frogs to behaviorally treat themselves. That's how they're describing it. Other treatment options that have been explored are fungicides. So those are medications that limit fungal growth. There's salt dosing, so increasing the salinity of the environment, selective breeding, and recently studies have also started to explore microbiome alterations, so the bacteria that grow on the animal's skin. Not only as a way to prevent infection or treat infection, but also looking at the impacts of infection on the microbiome over time and how that could lead to limits in growth or in survival long term. And so while all of these approaches seem promising, they're really difficult to implement at large ecological scales, which means that preventing the pathogen from spreading or being introduced in the first place is really our most effective strategy. And so while BSAL has not yet been detected in North America, regions with high salamander diversity would likely face the greatest ecological consequences if it were introduced. So Minnesota supports a really rich amphibian community. There's numerous salamander species that are distributed across our diverse wetlands and forest ecosystems. And this makes Minnesota ecologically valuable and also vulnerable to the emerging pathogen. So because of those reasons, our work was to identify the highest BSAL risk in Minnesota and then have some results that we can use for conversations around guiding surveillance and early detection efforts. Previous studies that have looked at BSAL and the risk of its introduction into the United States have combined the likelihood of a pathogen's introduction with those consequences if it were to become established. And they combine those to what they call this total or overall risk. So, for example, Riggles and some colleagues looked at a national spatial risk assessment, and they identified areas where the introduction potential and ecological impact were then really high. And that work was used then by folks that expanded on it and looked at regulatory actions and surveillance information. And so how could limiting the introduction of species that we know could carry this pathogen, how could that change the risk landscape and limit risk in certain areas? And those studies together showed that risk is not evenly distributed across the country. It's really concentrated in these parts of the Western US and along the Appalachian region where there's high levels of suitable habitat. And there's also high diversity of salamanders in those areas, which makes them highly vulnerable. But they acknowledge in their work that for conservation action and planning that we need finer scale analyses to be conducted, and that these studies need to be updated as we learn more about what species are susceptible. So our work is building on this foundation. We're applying this framework at a finer spatial scale to look at things at a county level within Minnesota. And though Minnesota is not our national hotspot, it's not lighting up here in the same way that the Appalachian region or the Western states are. It's, as I mentioned earlier, it supports diverse amphibian communities. We have extensive wetland habitats. And as the climate continues to change and things continue to warm, we might see changes in amphibian species preferences as well over time. We know that conservation planning and surveillance decisions are made at the state level as well. And so that's another reason why we wanted to look at this specifically within our state. So we followed a similar approach. We first assessed the introduction risk, which we're describing as the probability that BSAL enters Minnesota in the first place. Our current evidence suggests that the most likely pathway for the introduction is through pet trade. And through that route, infected animals can be transported. into the state. And so, as I mentioned before, it can be really challenging because some of those animals might not even be symptomatic. And so, if we were observing for lesions and didn't see anything, it doesn't necessarily mean that those animals are pathogen free. So the second component that we looked at is the consequence risk. So what are the ecological impacts if B-cell were introduced? We really looked at that through a couple of components. The first was the environmental suitability. We included that because the pathogen shows certain temperature conditions that it prefers to grow in. And then the second we looked at was species richness. And that was our way of measuring susceptible amphibian species and where they exist across the state, thinking about those consequences if the pathogen were introduced into their habitat. We went with species richness instead of abundance because there's some limitations in the way species are recorded and around surveillance. Some of these species are really elusive, and some areas haven't had as much intensive surveillance. But by combining what we know through these two components, we can start thinking about areas where the likelihood of an introduction and then also potential ecological impact would be highest. And that way we could prioritize those locations. So to approximate that introduction risk, we looked at a couple of indicators of the pet trade across the state. That included the location of pet stores. We obtained that through a database called Simply Analytics. It aggregates demographic, economic, business, and consumer data, and we compiled all of that. We also knew that there was a lot of activity happening outside of pet stores. There's a lot of trade occurring through reptile and amphibian shows. And so we then gathered data on what we could find about amphibian and reptile shows across the state. And we even visited a few ourselves as a lab. We didn't let them know that we were doing this for research, but we're just getting a sense of where their animals came from, what types of biosecurity practices or behaviors that they engaged in to make sure that their populations that they were breeding or showing were healthy, just to get a sense of what's happening in those arenas. So we then normalized these indicators and we compiled them to make a county level risk score. I'm showing that here. That risk score goes from 1 to 4. So we've normalized these indicators on a scale from 1 to 4. And that matches what the previous studies have done as well. We wanted to have a sense of how adding more granular data was comparing to the previous studies and if it was making sense given the new information that we had. So that's why we took this approach. And what this map shows here is that the counties with the highest introduction risk tend to cluster around the Twin Cities metro area. We're seeing Ramsey, Hennepin, and Dakota counties, which really isn't surprising given that these counties have high levels of human population densities, and they also have high concentration of pet stores and also trade activities. So, and then to get into the potential consequences if B-cell were introduced, we looked at those amphibian species in Minnesota that could act as hosts. To do this, we had Emily Banks, who is a veterinary student in my lab, conduct a literature search. So she went through knowing that there's been a lot of recent research around B-cell and amphibian species that might be susceptible. And that since the previous studies at the national level, since those have been published, a variety of different studies have come out. So we wanted to add that level of information in as well. We knew that some of them were susceptible and would be able to show clinical signs and have lesions if they were infected. But we also knew there were some that were acting as carriers and maybe would show very few clinical signs or none at all. And so the literature review is really important in being able to distinguish those and also update our knowledge. So we compiled observation records for those species that were identified as susceptible or carriers in the recent literature, along with what has been done in the past. And we took that list of species, I think we identified maybe close to 90, and we narrowed them down for those that exist in Minnesota. We then went through and looked at observation records. So we used several citizen science platforms. We used GBIF, iNaturalist, and HerpMapper, and we extended the range from 2016 to 2024. And what I'm showing here in this figure is that there's a number of observations for each of those potential hosts that are here in Minnesota. There are a few that are really dominating the data set. So here we're seeing the American toad appears quite frequently. These highly abundant and broadly distributed species can play an important role in disease dynamics, especially when they're in that carrier status, because they might not die and they can serve as a source of infection that then goes on and maintains it in the environment and spreads the pathogen through the ecosystem. And so here we are able to use this information and then map that to see where those locations are that are strongest, and then feed into our consequence risk. So using those host observations, we did just that. We mapped them, and we found that they were present in 85 out of 87 of our counties, which tells us that the potential hosts for B-cell are quite broadly distributed across the state. There's some counties that stood out to us with higher concentrations, and so we flagged those here. In particular, we see that St. Louis, Pine, Mille Lacs, they all show really high levels of species richness. And so thinking about what that means, right, those areas might face greater ecological consequences if B-cell were to be introduced, because then that pathogen would have more opportunities to establish and spread and potentially be maintained in those communities. And then that second part of the consequence risk is the environmental suitability. So our research, mostly laboratory studies, they have shown that B-cell growth occurs best at 15 degrees Celsius, and that it doesn't tolerate temperatures above 25 degrees Celsius well at all. So we went through and used that information to evaluate how suitable Minnesota's climate might be. We used the temperature data from the PRISM climate group, and we looked at mean annual air temperature and mean July air temperature across our counties. And then we combined those temperature variables to look at how closely local climate conditions match the thermal range that those studies have shown that B-cell is able to persist. And so this map is showing that resulting environmental suitability score. Overall, we see that southern Minnesota appears to be more suitable, where temperatures fall closer to that optimal range, and that the northern counties appear less suitable. When we combine this environmental suitability with our host availability, and then also our introduction risk, we can then get that bigger picture of B-cell establishment and spread. So we combine these components that we just discussed. We looked at that host availability and that environmental suitability, and we integrated those two factors. We generated a county level consequence risk for Minnesota. And so this map is highlighting that where the conditions align. Several counties then stood out to us here once we combined those scores, and that included Pine, Wright, and Dakota counties. So these areas, again, they might represent that B-cell establishment and spread, not necessarily introduction yet. But once we had that introduction score and we added it, we used what previous studies have used called an unweighted linear combination. And so that means that these layers are combined, and then the highest relative risk starts to appear in these areas where the higher introduction likelihood coincide with those areas of that higher consequence. And so that's what we're showing here. What stood out to us is that in Minnesota, the overlap is most evident in that southeastern part of the state and kind of concentrated around the Twin Cities metro area. So to support early detection, we established a statewide surveillance network that we're calling Minnesota Snaps. Minnesota SNAP stands for the Minnesota Student Network for Amphibian Pathogen Surveillance. The network is a combination of colleges, universities, zoos, aquariums, and nature centers across Minnesota that have been invited to sign up. It is not exclusively for folks who've received invitations. We are open to working with folks or groups of folks who want to go out in the field or are already out in the field, maybe doing things like AIS surveillance in general, who want to also swab amphibians and send them to the lab. Through this program, the participants get all of the sampling equipment that they would need shipped to them and a cooler. And the postage is also paid for. So when folks go out and collect their samples, they can then send them back to the lab. And the project pays for all of that. There are training materials as well available for folks. So if they're interested and they sign up for the program, they get invited to a training and understand what the protocol is and what biosecurity practices are so that folks don't accidentally spread BSAL or BD to other locations. If it were present in the ecosystem, you can imagine doing surveillance and then spreading the pathogen that you're trying to prevent spread of would be very unfortunate. So yeah, they're playing a critical role in that broader surveillance. And if folks are interested, we can share ways for you all to learn more about it. This is my lab, who's been carrying out surveillance efforts in the field. This was them out in the summer of 2025. When we collect the data, we share it through the amphibian disease portal, and I'll share more about that. But those data go into citizen science platforms. So not only are the species that we're identifying being recorded and shared out in the citizen science platforms that we've used to inform our models, but we're also contributing out to other initiatives that are occurring across the globe and across the country and the state. Our lab is really focusing on training students and also building capacity for pathogen surveillance, highlighting how to safely capture amphibians, how to do non-lethal skin swabs. So all of the animals the goal is that they are all alive. There are protocols if animals are injured or showing symptoms of BSAL or BD infection and what to do in those cases. And what we're doing in Minnesota with the Minnesota SNAPs fits into a larger national effort. It's part of the Student Network for Amphibian Pathogen Surveillance. That's the national SNAP's name. Across that network, they have about 60 institutions who are participating and overall have collected more than 2,500, I guess close to 2,600 amphibian samples to date. So each of these spots that I'm showing here on the map shows where these partner institutions are and help build out that broader understanding of where the pathogens occur and where new introductions might be emerging. So we're swabbing amphibians looking for BSAL, but we're also collecting data and testing for BD as well, just to understand what's happening in those environments over time and can serve a greater purpose as well at a broader, more scientific level. As I mentioned, these samples and what we collect within our lab go into this amphibian disease portal. It's a centralized database, and researchers around the world can use it, and they also contribute to it as well. And so the portal allows scientists, they can query their data, they can start to visualize patterns, they can then look at areas where there's higher levels of activity. And the work that we did in Minnesota SNAPs in 2024 and 2025, we submitted information on over 300 amphibian samples. So BD was detected in some locations. There was a prevalence of up to 69%. That means that 69% of the animals that were swabbed in certain locations were positive. And we also, in other locations around the same time, found that no animals were positive. And so there's a high level of variability and some complex environmental factors that lead to the presence of BD across the state. And there, Carrie Beers, which is one of the students I noted as part of our larger project team, is working with Jennifer Lamb at St. Cloud State University, and she's asking a variety of questions around this. So what are the drivers? How is infection levels different in different species, and what are the likely driving factors? So one good thing to note is the B-cell has not been detected in Minnesota through our surveillance efforts. To date, it also has not been detected or reported in North America, which I mentioned earlier in this talk. And so we're hoping to keep it that way through these surveillance efforts. And so one way we engage colleges and university instructors to participate in the program is through something called a course-based undergraduate research experience. And these are often called cures. And so within these courses, within courses where instructors participate, the students then engage in research projects, and the data they collect are used to answer questions where there's really no known outcome yet, right? So a lot of times in class, students are given problems, they're asked to solve them, and the solution is already readily available. Well, in here, we're trying to get students to think about what their hypothesis is given previously collected data. Or if they're not at that level, maybe the instructor gives them the hypothesis and they think about collecting the data and being able to answer whether their hypothesis was correct or not. And so there's a variety of different levels that instructors can engage through these cures and connect with other researchers who are using this within their classrooms. The idea is that folks are building research skills through classes, they're practicing science communication, working with those, as I said, real scientific questions, and they don't have to do that outside of their course load. Some folks who get involved in research have done it through volunteering, and this is making it more accessible and more available, while also trying to collect more information on what's happening with amphibians and amphibian diseases. It is important to note that these cures cover a range of topics. Broadly, they are used at undergraduate institutions, and our contributions have been related to amphibians and amphibian diseases. And so that's been part of the work that we've carried out, is to develop a SNAPS focused cure that is focused on how do we access and visualize our surveillance data. And so Jennifer Lamb, one of my co-leads of the project, has built out a Shiny app where instructors who want to be part of the cure maybe have noted in the past that accessing data from the amphibian disease portal and getting it accessible to students who maybe don't regularly engage in data visualization intensely has been a challenge in the past. And so we're trying to overcome some of those barriers and also enable them to still ask questions about spatial distribution and intensity and things like that. So they can go in and they can upload the data that they've collected and bring in samples that have been reported from other locations and start to visualize that here in the app. They can look at the geographical location, those diagnostic results, what species were sampled, and then again, it links out into these larger surveillance systems. Folks can communicate. So I'll just go ahead and close out the talk by highlighting a few directions where our work is moving. And our next phase of the work, we are focusing on working with state and regional agencies. So we're thinking about taking our risk models and moving it towards practical B-cell response planning. So things like, what are your conservation priorities? We want to strengthen our surveillance networks and develop contingency plans so that if B-cell were detected, the communication networks have been built out, the potential scenarios or what-ifs have been explored, and the trade-offs have been thought of, and what next steps need to be to really implement things on the ground. We're also continuing to sample amphibians and refine our understanding of their distribution. and their species richness. And that can help us as we move through and aim to improve the accuracy of our risk assessments. When we do surveillance, when we have negative results and the pathogen has not been detected, that can still be used as information to decrease our uncertainty, right? Our diagnostic tests aren't perfect, and we're also not able to sample every amphibian in the ecosystem. And so by taking our effort and even our negative results, we can start to get a better picture of at least refining our understanding of the probability that it's truly not there. And there have been some studies who've explored that more explicitly at the national level. We also want to look at using eDNA or environmental DNA to better understand habitats that are used by eastern newts. And we are specifically looking at eastern newts because they are a species here in Minnesota that are particularly susceptible to the impacts of BSAL, and they're also highly elusive. So if we can utilize eDNA to give us information about where active eastern newt habitats might be, it can help us with our sampling. And also we're exploring using eDNA for identifying BSAL and BT as well. And there's been a lot of interest around eDNA and its limitations. And so contributing our efforts in understanding and those limitations, and probably also some frustration that goes along with that into the larger conversation. So that's our goals there. With that, I would like to acknowledge our funding organization. So this work has been supported by NASERC and the Minnesota Aquatic Invasive Species Research Center and the Environment and Natural Resource Trust Fund. And I also want to thank folks who supported my lab's work sampling out through the Twin Cities. We worked with the Three Rivers Park District, Ramsey County Parks and Rec, and I want to thank our Minnesota SNAPS partners, the folks who've expressed engagement, those who've been able to go out and sample to date, and then the National SNAPS program for supporting us with organized testing and training resources and things like that. And with that, I would like to open it up to you all and hear what's on your mind.
Wonderful. Thank you so much, Amy. I am going to take over screen share real quick. So as I mentioned at the opening, we have a tool called Slido that we use to help moderate Q&A. It's a really great tool because you get to upvote questions. So it helps us bring the questions that folks are most interested in answers to up to the top. So you can scan that QR code showing on the screen now, and it will bring you directly to the Q&A. You can go to slido.com and enter the event code Amphibians and join that way. And if the tool isn't working for you or you prefer not to use it and you do end up putting your question into the chat, that's okay. Michelle is going to grab it and put it into Slido on your behalf, and that way I'll see it as I'm working through the questions that come in. So I see we've already got some flowing in, so feel free to keep the questions coming. And Amy, I'm just going to get started up at the top of the list right now. And the first question that I see here is wondering what you know about how equipment like waders and dip nets need to be sanitized after they're used to help prevent the spread of like Chytrid fungus, or I'm sure other diseases as well.
Yeah, that's a great question. So, yeah, that's something we certainly want to prevent when we go out and do our sampling. And that's one of the contributions that we're making from this project as well. So we've generated educational materials, and one of those is a video on how to decontaminate your equipment. And so generally, the recommendation is to use Vicron, which is a disinfectant that you can order. It's available on like Amazon, or you can search it up, and it tells you in the video how to dilute it in the field, which can be a challenge. Or you can also use bleach. And so the other thing I'll add is that we have a permit through the DNR, an umbrella permit that supports folks out sampling. But if you go out in specific parks or locations, you'll also need to check and see if you need to get a local level permit. Within that, some organizations require that you do disinfection protocols that they've developed. And so I would just inquire about that and make sure that you're following the most stringent disinfection protocol. on the Macer website. I'm not sure. Can I add in the chat, Megan? Can I add
Yep,
things in the
yep,
chat?
absolutely. Yeah, just make sure you
Okay.
pick everyone and who you're chatting to, and it should go out for everyone to see.
Okay, sure.
The
So
default might be to hosts and panelists. So
yeah, so Macer subproject 54 Chytrid. In here we have training. If you search that up in there, there's training materials, there's the video. There are results from our previous studies around what we found with our B cell surveillance. I'm gonna put this in the chat. If you can't find the information that you're looking for, feel free to shoot me an email too. I'd be happy to direct you to those locations or connect you with national snaps or enroll you in Minnesota snaps. All those things are available. But Vicron and bleach, and then in the video, we walk you through how you can implement that in the field where, thinking beforehand, like bringing a giant tub and making sure that you have your brush and making sure you're organizing things in ways has been really helpful for folks.
great.
Mhm.
All right. Next question, working down the list. Have you considered exploring the actual risks of the pet trade by visiting pet stores that sell amphibians and testing for BD and B cell there?
Yeah, so that's another part of our phase two. I didn't explicitly acknowledge that because there's some challenges there that we're thinking about how we overcome. We've connected with folks at the DNR who've been working in aquarium trade for a while, and they had some really great thoughts on where to go around this topic. But our main challenge is data privacy. So if we go out and we do surveillance and we identify that the animals are carrying BD, some pet stores maybe don't want that information released. And when we're doing research at a public institution, right, our goal is to share our knowledge. So how can we do that while maintaining anonymity and making sure that our results are also valuable and important and interpretable is kind of what we're thinking about now. We have some surveillance funds that we can use to do that. We're using DNR connections and ideas that they've had to start mapping out what that might look like, that data privacy piece of it. And I think a optimistic thing to note is that other studies around the US have done surveys of pet stores that sell amphibians, and have asked, would you be interested in either paying to be part of a biosecurity maybe program where we test your animals and we help you think about how to keep them safe. And overwhelmingly, the support was there. So we don't expect that the pet stores will say no. We just want to make sure that we're approaching it in the best way and realize that folks have the right to say no, and we might face some challenges there. And we're thinking about doing this at a broad scale. So yes, I love that idea. I love that support, thinking about it and thinking about what that really means to implement.
Right. Okay, next question. It looks like there's a few people who are at least interested in getting involved with Minnesota Snaps, and they're wondering what the best way to do that is.
Yeah. So if you send me an email, this car0603@umn.edu, I will connect you with the folks who will get you all of the registration information. And I'll also follow up myself and Jennifer Lamb to help answer any questions that you have about the permitting process. So if you are interested, please do not hesitate. If you are interested, but not exactly sure if you will be able to carry out this summer, that's okay. Like folks who just want more information or think they might, connect all the time. We just ask that if you aren't using your resources that we send you, your sampling kits, that you just send them back to us.
Great, perfect. Okay, next we're into some more specific questions about the fungus. And the first one is wondering if there's a dormant life stage where they can overwinter in cold temperatures.
Yeah, so they can overwinter in cold temperatures, and there's actually a couple of mechanisms that folks think that the pathogen utilizes. So one thing that's really interesting is that this pathogen, as I mentioned, it has that critical part of the life cycle within the skin, and it really prefers the keratinized tissue. So when we're thinking about tadpoles, really the only keratinized tissue they have is around the mouth. The rest of their skin is not yet keratinized. And so what's really interesting is folks have gone out and they've sampled tadpoles and they find really high levels of BD in these tadpoles. And the hypothesis is that these tadpoles, as they go through metamorphosis, if it's not killing the tadpoles because it's really only around the mouth, if it's not affecting their development, it's not, distributed all over the body, they tend to have persistent infections. And then they grow into juveniles and are potentially these, even if they're a species that are symptomatic, if they're infected as adults, they might become asymptomatic carriers. So that might be one route. If they're overwintering within a tadpole, that could be a source in the spring. Another thought is that whenever the zoospores are frozen, if it is a quick freeze, it's more likely to be viable in spring when it thaws. If it's like a slow freeze or what we've been kind of experiencing are these freezes, thaws, it's less likely to. So these volatile kind of springs and winters might actually be really great for decreasing BD prevalence. But yeah, they can survive. A lot of folks wonder how amphibians overwinter, and like terrestrial amphibians will overwinter. They bury under the frost line. We might see species that bury at the bottom of the pond. They might go into crevices and overwinter there. And some amphibians have this ability to utilize glucose as like an antifreeze within their body. And so they actually can just withstand freezing temperatures. And so I'm sure there's studies out there that I haven't dug in too deeply as to whether infected and asymptomatic, so animals that aren't symptomatic but then are also infected, if they have a difference in their survival of overwintering. I haven't seen anything around that. It doesn't mean it doesn't exist. It just means I haven't dug into it. But that's kind of largely how we see the pathogen persisting in cold environments over time.
Yeah. Wow.
Yeah, it's interesting.
Yeah. Okay, next one, wondering a little bit more. Where did the BD fungi originally come from? And do you know where it was first identified and then like how it spread from there?
Yeah, there's a large story and a lot of phylogenetic work that has been done. And my understanding is they've traced it back to populations in Asia, and that there are some populations there that are not affected, and that it then spread through global pet trade, and that folks were seeing large population declines, and most notably in Central America. And when that was happening, folks started communicating to each other. Scientists who, like herpetologists across the world, were starting to connect and talk to each other about it. And that's how they found there was this global pandemic occurring and that it was affecting amphibian populations. There are maps out there that kind of trace back the route of spread or hypothesized routes of spread with different lineages. I'm not able to recreate that for you verbally, but essentially to answer your question, they think that it came from populations that were relatively resistant in Asia.
Okay, next question is moving on to some regulatory and education efforts. So first, wondering if there have been any discussions around legislation in Minnesota around pet and reptile laws. This says if not, but maybe in addition, if there's efforts about education around like shelters or herp societies and hobbyists to prevent pet release that could lead to spread.
Yeah, pet release is a big deal. Okay, I'll answer the first question first, which is about legislation. And so there aren't laws that prohibit the importation of wildlife diseases, but the Lacey Act does prohibit the importation of injurious species. And so in 2016, like 201 or some large, about 200 species were prohibited from importation and interstate movement in the United States. And that was due to the decimated populations in Europe, right? So those fire salamanders that I was telling you about, right? So around that time, they limited the importation and movement of 201. At that point in time, like what we think are likely susceptible species. Since then, and I got a bit into the research that has been done with the national risk map. Greer, who is one of our collaborators on this project, was explicitly looking at, well, we're getting more information about species that are susceptible through either experimental studies, what folks have seen in the wild otherwise, maybe captive animals. what happens if we expand that those 201 injurious species, that definition, into these additional species? Are we really making an impact on risk? And the answer, even if it was one, there's probably a lot of us who are like, yes, yes, expand it. But having that data and that evidence there is really helpful. And so in 2025, they added like 16 new species to that list. And so that's really where regulation is coming in. I would also note that it's really hard to regulate things you can't see or you don't have evidence of. And there is a lot of global pet trade that we just aren't aware of. And so that is something that um, has been highlighted in this study for me as well. We went to those expos, we chatted with people. Folks seem really aware and concerned about managing the health of their populations. And also, it's really hard to track online sales. It's really hard to really get your hands around where animals are moving when. And I can imagine that's also happening internationally still as well. Not to be, not to scare anyone, but it's just part of the world that we live in, right? And so legislation helps. I think raising awareness helps. Certainly that limits the potential risk, but it's not completely zero, which is why we're still out doing the surveillance that we are, and we're still having conversations with folks. So bringing that to the question about pet trade, um, sorry, Megan, I think it was about limiting people dumping their aquariums.
Yeah, yeah, if there were efforts for like education for discouraging or preventing pet release.
Yeah, and so I know the DNR has done a lot with the aquarium pet trade. I know that the Minnesota SNAPs, and there is a BSAL task force that focuses on bringing folks together from a variety of different organizations and thinking about how do we limit introduction and how do we work together if there were an outbreak and how do we generate materials for people. I know there's a lot of work around there for increased awareness. I think at the local level, that question about how do we work with pet stores and how do we get information out through those pet trades that we do know about is probably our most effective route at the local level. And so, in addition to working with the pet stores and sampling their animals, I think the next reasonable step would be how do we also communicate to new pet owners what the steps are that they can take if they no longer want these animals. And there are a variety of resources where folks take in these animals and try to find them new homes or keep them as their forever home as well. And our veterinary student is really active in those boards as well, because they also give us a good understanding of what species are out there at the local level.
Yeah, great. I know that's a problem across many different taxa in aquatic invasive species prevention. And
Yep.
all right. Next question, wondering about if the infections can be treated in individuals or at a population level in an area.
Yeah. So there are those mechanisms that I mentioned, or those tools that folks have tried to use. So those are things like antifungals or increasing salinity levels, or um, think about like zoos that are engaged in captive management or captive breeding so that they could reintroduce populations or individuals to establish new populations. That can be challenging, especially if the threat still persists. And for BD, it's really challenging once the pathogen is in the environment to eradicate it. So, in captive populations, those things have been successful. Those frog spas or frog saunas have also been successful for population levels and individuals. I think when we start thinking about it at a landscape level, those things start to get more challenging. And then there's also trade offs with if we are using fungicides or some sort of chemical for prescribed treatment that we don't always necessarily know what the secondary effects of that could be or what could happen to our non-target species. And so I think within that, there's always risks being considered. And in general, kind of the consensus is that these tools are here, but we're still really looking for something that's effective more broadly.
Sure. Yeah.
Yeah.
Okay, so going back to the maps that you were working on, the next question I was wondering about as you were combining the risk factors to kind of layer and create that final map, were you weighing any of the variables differently than others? And if so, how'd you determine that?
No, we aren't doing that. So we're using this unweighted linear combination, and that is everything is treated as equal. And so I think that when we get into that, we really need more information about how introduction and spread occurs on the landscape in real life before we can start thinking about how we weigh those. We took that approach because that was what folks had done at the national level. But I am not against having stakeholder feedback and modifying our local level maps to weigh things differently based on what we understand is happening at our local level. So the answer to that is no, not at this point. This is kind of our starting point and baseline that we can use as we continue to do surveillance and update, decrease that uncertainty, and also maybe start tweaking some of those factors. But no, and I think another um, point within that is it makes it a little bit more easy to interpret how these factors are layering in on each other and creating these spatial patterns, knowing that it's pretty transparent that these things are equally weighted. So at this point, we kind of see it as a limitation and in some ways a communication strength.
Sure.
Yeah.
All right. Um, Next question, wondering, can these diseases affect other herps like turtles or reptiles?
Yeah, fortunately, they do not. They are, from what we understand, strictly infecting amphibians, though research has shown that crustaceans or crayfish in particular have had BD in their shells and in their GI tracts. And it has been hypothesized that potentially they're serving as reservoirs or hosts. So maybe they're carrying BD unaffected from what we understand. We can recover it from the shells in the GI tract, but we aren't seeing any real lesions that look like they are leading to any negative health consequences. We can do that under the microscopes. We can take their GI tract and like section in these tiny pieces and then look at it and see how the tissue looks. And so the thought is that maybe they're carriers. And that then opens up the dialogue that maybe there's more carrier species out there. We haven't seen significant population impacts from this. So at this point, the answer is, from what I understand, no. But we're keeping our eye on crayfish and thinking about that and the potential impacts for like invasive crayfish and what does that then mean in some environments. So like invasives and invasives interacting, right?
Yeah, it sounds like it has the potential to create a whole new risk map.
Yes, certainly, certainly.
Great. All right. Next question. When you were pulling data about the prevalence of various amphibians across Minnesota, they're wondering if the Minnesota frog and toad survey data was also one of the resources that you looked to.
Yes. So anything that was reported in GBIF, which collects a variety of different surveys, I don't know if that one specifically was included within there. My understanding was GBIF was pretty comprehensive. That iNaturalist or Herp Mapper, we went through and did our best to try to remove what we perceive to be duplicate records. And the veterinary student in my lab can identify frogs by call. And so she was listening to those that also had frog calls and was removing those if they were noted as the incorrect species. But I'm not exactly sure, but I can go back and check our GBIF data, or you could check what was in the GBIF data if I'm too slow in responding to you and you shoot me an email. But yeah, we try to get our hands on all of that. I do know there's organizations who are doing sampling. And if they're not sharing those through those portals, that they have offered them to us in the past. And if you have any of that, I would be happy to take it.
Right. All right. I think we'll go maybe one more question here. Last one is wondering if you looked at what the most or what species are common in pet trade that might be carrying the disease. So are there any that you recognize that are common that might be the highest risk species?
Oh, that's a great question. And I don't know this to be fact, but those tree frogs tend to be really popular. I see those traded a lot. Those have been noted as a carrier or susceptible, and have shown up in our surveys quite a bit. So we know that those susceptible species are out on the landscape broadly, and we also see them in those pet stores. So that's the first one that comes to my mind. And yeah, probably a risk. American bullfrog, who is quite broadly distributed, not really sold often, but something else we're keeping an eye on because they're asymptomatic and could be infected, and we people wouldn't report it, right? We would only know if we went out sampling. So that's
Yeah.
those two things I think are our highest risks.
Great. Well,
Mhm.
thank you so much, Amy. That brought us right to 2 o'clock. I just have a couple of parting messages for folks. So, thanks to everyone for joining us today to hear from Amy. And thank you so much, Amy, for joining us to share all of this great information. It was fun to have such a new and different topic be a part of the webinar series. We really appreciate that.
Great.
The webinar was recorded, so you can watch our YouTube channel. You can get to at z.umn.edu/AISTube. And the recording will go live there, but give us a little bit of time because we're going to transcribe it before it goes up. So you'll get an email once it's live and ready to go. And there should be a survey link that pops up when you leave. And we'd really appreciate if you took the time to click that link and answer a few questions about the survey, or sorry, about the webinar as well. Amy put her email into chat. It is also showing on the screen right now. So if you were interested in getting involved and wanted to reach out, that's the email address to reach her at. And if you have questions for us as webinar hosts, other webinars, things like that, our email is showing on the screen as well, AIS Detectors at umn.edu. That's all that we had for you today. But we will look forward. Hopefully, you'll be joining us for our next webinar. We host them every other month. And now I'm going to forget what the next topic is off the top of my head. Give me one quick moment. But you should be getting reminders that these are um, coming up as well if you have joined us for a webinar before. Oh, it's our genetic biocontrol talk next featuring Mike Szymanski. So another fun, interesting technology type talk. So we'll hope to see you at that one in the future. Thanks, everyone.